Okay, see, you people are going to have a test, right? I think maths and physics portions are clear to you. Don't you need biology topics, right? Will you be able to learn Unit 8, Biology in Human Welfare, 3 chapters. Human Health, Strategies, Microbes in Human Welfare. Okay, will that be fine? That whole unit, chapter 8, chapter 9 and chapter 10. Okay, so shall we fix that? Biology in human welfare. When is the test? Did they give you a date? Did they give you a date? Anyone? After 15th, after July 15th. Okay, so I think it's easy, right? Chapter 8, 9 and 10, 3 chapters for you. Okay, physics also 3 chapters, right? Yes or no? Okay, so we'll fix that. I'll put it in the group. Learn chapter 8, chapter 9, and chapter 10 for your exam. Be prepared. Okay, I don't know when the date is. Anyway, be prepared. So, shall we learn something quite different today? Ecology, a bit of ecology. A very simple chapter we'll take today. Chapter 15. Okay, chapter 15, biodiversity and conservation. Okay, biodiversity and conservation. So please open your textbook, and after that we'll do the questions. I think yesterday also I sent a few questions up till question number 80. We'll discuss that also after this. Got it? Hope you have done it. Up till how many questions did we discuss from biotechnology applications? How many did we discuss? Up till 30, 30, right? Did we discuss up till question number 30, 30? Just check and tell me. I think we have to discuss from question number 31. Okay, so we'll discuss from question number 31 to question number 80. Hope all of you have the questions with you. And Surya had sent me the answers of it. Okay, so shall we start? Chapter 15, Biodiversity and Conservation. Very simple chapter. Just go through what you have to learn. We are talking about biodiversity and we are talking about the conservation aspects of it. So this is the chapter. Before we start the chapter, turn the pages and see what all you have to learn. They have given the word biodiversity. Okay, and they've given a few examples of that biodiversity that exists. And the next heading is how many species are there on the earth and how many are there in India. Okay, and if you turn the page, take page number 260, you're going to see 3 pie diagrams. Do you see it? The happy news is you're supposed to learn that. The 3 pie diagrams. Did all of you see it? Invertebrates, vertebrates, and plants. Okay, then you can see patterns of diversity. Okay, in patterns of diversity, latitudinal gradient, species area relationship. Today we will complete up to latitudinal gradient. That is our plan. Okay, and then you can see importance of species diversity in the ecosystem, loss of biodiversity, causes of biodiversity loss, why should we conserve biodiversity, and how do we conserve biodiversity. So it's like a question answer format. Okay, the heading you have to read, and that paragraph coming after that will be the answer for that, right? So this whole lesson has been arranged in the form of a question answer format. Got it? So shall we start? So first one, they have given some examples of biodiversity. If you look around you, you find there are a huge number of animals, huge number of plants, insects, right? The variety is just stunning. You look around you and you are amazed at this beautiful biodiversity. But sadly, because of habitat loss, because of killing of animals, over exploitation of these plants and animals, they are becoming extinct. That is the fact. Yeah? That's a very sad fact that we are seeing around us, right? You can see they have given a few numbers there. Please underline those numbers right now. How many species of ants are present? Find out how many species of ants. Right in the first paragraph, you're going to see it. Underline it right now and put the number in the chat box if you have seen it. How many species? Navneet has seen it. 20,000, right? Species of ants. 3 lakh species of beetles, right? 28,000 species of fishes and 20,000 species of orchids in the world. Okay, this is the number that is found around the world. Hope it's clear. So underline it, learn it right now. After that, the concept of biodiversity, the person who popularized this word. Okay, the person who popularized the word biodiversity, his name is Edward Wilson. Okay, the person who popularized the word biodiversity is Edward Wilson. Hope you understood this, right? So, this scientist, he was actually learning ant communications. He was a specialist. He was learning about ants, how ants communicate with each other. He was blind in one eye. Okay, when he was when he was fishing, the fishing hook accidentally came and hit his eye, and he was blind in one eye. But he started focusing on very small things after that, right? And his favorite topic of study was ant communications. Got it? So Edward Wilson, he was learning about the populations, he was learning about ant communications, and he was the one who popularized the word biodiversity. And what is biodiversity actually? It is the heterogeneity that you find around. Okay, the heterogeneity that you find around. Just underline that word heterogeneity from your textbook. Heterogeneity that you find around yourself, you call that biodiversity. Please underline that. And this heterogeneity is not only at the species level. He said that according to Edward Wilson, biodiversity exists in 3 levels. Okay, so the next thing, biodiversity exists in 3 levels according to Edward Wilson. What are those 3 levels? The first, the basic level, that is the species level. Okay, at the species level, or you'll write it like this, okay, in your textbook. Okay, first one, genetic level, the genetic level. Okay, then the species level, and then the ecosystem level. Okay, so there are 3 levels, ecosystem or the ecological diversity. Okay, so 3 levels. Genetic, you can write it as genetic diversity. Okay, so this will be species diversity, right? And this will be ecosystem, or you can write ecological diversity. Okay, so once again, according to Edward Wilson, right, he popularized this word biodiversity, and according to him, there are 3 levels of biodiversity. What are the 3 levels once again? One is the genetic level, or you can call it genetic diversity, right? Second one, species level. So what will you call it? Species diversity. And the last level is the ecosystem level, or you can call it the ecological diversity. Got it? So when we will look at the examples, and the examples are the one that is asked for the exam. Genetic level. Look at that example. They have given Rauwolfia vomitora. Please, did you see it, everybody? In the Himalayan regions, there's a medicinal plant. Okay, its name is Rauwolfia vomitora. Did you see that? It's found in the Himalayan regions. And there's a special drug that is extracted from that plant, and it's called reserpine. Okay, the drug is called reserpine. Okay, and you can see that there's a huge genetic diversity for this plant. Did everybody see that example? Give me a yes if you have done it, if you have seen it. BB has seen it. Everybody. Devika. Okay, so what is the name of the plant? Rauwolfia vomitora. Where will you find it? In the Himalayan regions, right? What about that Rauwolfia vomitora? You find a number of, there's a huge genetic diversity for that plant. Okay, what is that active ingredient that you extract? It's called reserpine. The active ingredient that you obtain is reserpine. Hope it's clear, dear children. Okay, and they have given different types or genetically different types of strains of rice. How many strains of rice will you find? 50,000 genetically different strains of rice. These numbers you are supposed to by heart. Okay, and 1,000 varieties of mangoes you're going to find. Did you see these two? 50,000 genetically different strains of rice, 1,000 different genetically different strains of mango you're going to find. Okay, mangoes, you know, right? Different varieties of mangoes are present. Alfonso, Jahangir, Muandan, right? Chappikudian, right? Our own native varieties, Malgova, Sundari, right? Lots and lots of varieties of mangoes you'll find. There are nearly 1,000 varieties of mangoes that you're going to find. That is in India, okay? In India alone. You're talking about India. So these numbers are for India alone. Hope you understood this, right? By heart these numbers. So all these things come under genetic level. So they will give you 1,000 varieties of mangoes are found. Okay, 1,000 varieties of mangoes are found. Which level of biodiversity are we talking about? What will be your answer? This is a question that they have asked before. They'll give you 1,000 varieties of mangoes are found. Which level of biodiversity are we talking about? Which level? Genetic level. Yes, Surya, genetic level. Felina, Devika, we are talking about the genetic level. Shall we come to the species level now? If you look at Western Ghats, okay, and compare it with Eastern Ghats, you will find more species of amphibians in the Western Ghats than in the Eastern Ghats. Okay, you know that Western Ghats is a biodiversity hotspot region. Lots of species are present. So one example is amphibians. Okay, the number of species of amphibians that is found in Western Ghats, it will be more than Eastern Ghats. Okay, Western Ghats, you're familiar. They are trying to protect those Western Ghat region. There has been a loss of biodiversity, loss of habitat in the Western Ghat region. You might be familiar with the Madhav Gadgil report. Are you? Madhav Gadgil report. Whenever there are floods, Madhav Gadgil report is the topic of discussion. Are you familiar with that report, dear children? Madhav Gadgil report. Nobody is familiar? Whenever there are floods, in Madhav Gadgil's report, it is specifically said how to protect those biodiversity in the Western Ghat region, right? During floods, you have seen it in Wayanad, right? And all those regions due to excessive mining, destruction of those trees there, the whole region has collapsed, right? And this rainy season also, we don't know what is going to happen. Yes. So species diversity, right? Species level diversity. Western Ghats, there's more amphibian species when you compare it with Eastern Ghats. That's just one example, okay? Coming to the third level of biodiversity, that's called ecosystem level, or you can just call it ecological diversity. Just take India as the example, right? From there are different geographical areas that you find. There can be beaches, right? Plateaus, plains, desert regions, mountainous regions, right? Lots and lots of biodiversity, lots and lots of ecological biodiversity you can see. Is this fine? So just take India as the example. All types of terrains are found in India. From hot deserts to snow-capped mountains, we have everything, right? So that will represent ecosystem level of biodiversity or ecological biodiversity. Hope it's clear. But if you take some other countries, like they've given an example, Scandinavian country like Norway, the whole country is alike, the whole physiology is alike, right? But in India, you find a number of different types of ecosystems. Hope it's fine, dear children. 3 levels. Is it okay? And this is a 2-mark question. Okay, this usually comes for 2 marks. So just note it down. They might ask you maybe 2 levels, okay, to explain any 2 levels of biodiversity. Okay, question can be specific. They can ask you genetic level or species level. The examples are super important. So please note it down. So do one thing, just quickly read biodiversity. Just quickly read up till that, see if it's fine. Underline these points in your textbook and write the mark. Do it right now. And after you're finished, just tell me. Okay, is it done, children? Is it done? Have you finished reading? Hope you are reading aloud. Hope the examples are clear to you. Right, Devika, Felina, good. What about the rest of you? Read it fast, very fast. Just to see whether you have understood it. Okay, Navani, Amulya, good. Okay, next one, next term. It's not there in your textbook, but just note it down. Okay, Ria, Aisha. It's not there in your textbook, but just make a note of it somewhere. Okay, it's called keystone species. Okay, keystone species. This is a very important concept. Do you know what a keystone is? Anyone? Keystone? See, if you build a big arch like this, the stone that is found here, okay, right in the center of that arch, you call it the keystone. What if you remove the stone? This whole thing will collapse, right? So there are some species which are very, very important in the ecosystem. They are very few in number, but their impact is huge. In humans also, there are some people like that, right? Even though it's just a single person, their thoughts, their effects will affect the whole society. Don't you think so? Right? So there are some keystone species. They are very less in number, but still, they influence the ecosystem very much. Okay. For example, I'll give you an example of a keystone species, that is jaguar. Okay. A jaguar. You're familiar with jaguar, right? Not the car I'm talking about. The animal, right? The speciality of jaguar is it eats nearly 87 different species. Okay, it eats nearly 87 different species. Okay, and if jaguar becomes extinct, just imagine jaguar has become extinct. What will happen is that entire ecosystem will collapse, because all these species will increase in huge number. There'll be competition, right? And it can lead to the extinction of a number of different species. Okay, so it can lead to the extinction of all these different types of species. Got it? Once again, jaguar, it eats a number of different species, right? And if this becomes extinct, it can lead to the extinction of a number of different species. So such a species, you call it a keystone species. Even though jaguar is very few in number, they will affect the entire ecosystem. Okay, now there's another one, sea otters. Okay, sea otters can also be called a keystone species. Even they affect the aquatic ecosystem in an enormous manner. Okay, so such species are called keystone species. This is just for your entrance, it's not there for your board examination, right? So the next one, the next concept, there's a question asked in your textbook, right? How many species are there on the earth and how many are there in India, right? So there's a published report according to IUCN. This is a very famous organization, right? IUCN is International Union for Conservation of Nature and Natural Resources. Okay, we'll just write it down. International Union for Conservation of Nature and Natural Resources. Okay, earlier the definition was the expansion was only up till here, International Union for Conservation of Nature, but they have added natural resources also along to the list. Okay, so according to IUCN, how many species are present in the world? Just find out and tell me. Find out from that paragraph and tell me, according to IUCN, how many species are present on the earth? This number you're supposed to by heart. Find it out fast. Let me see who finds that number fast. Page number 259. Surya has found it. Okay, 1.5 million, more than, right? You can just say 1.4 to 1.5 million species are present on the earth, according to IUCN. Okay, there was another scientist, an ecologist. His name was Robert May. Okay, so find out, according to Robert May, how many species are present in the earth. Did you find it? Hey, Surya. From where did you get that number? According to Robert May, Surya, did you see? 7 million, right? 7 million species are present. These two numbers you're supposed to by heart. Got it? And the problem with finding out biodiversity is, see, before you even discover something, some animals are becoming extinct, right? So recording biodiversity is a big problem these days. Most of them are hidden deep of frogs. We are still in the process. But these are the authentic numbers. Yes, Navani, it's getting stuck. Okay. Is it fine now? Is it okay? So hope this is clear. So from that paragraph, these are the two things you should learn. Okay, just two things. Shall we proceed to the next one? You can see 3 pie diagrams. Turn the page, everybody. You can see 3 pie diagrams there. And you're supposed to by heart those things. We'll take first, you can see invertebrates. Right? You know what are the invertebrates? Insects, right? Mollusca, crustaceans, all those things will come under invertebrates. So those numbers you're supposed to by heart. Which one is more? See, if you take plants and animals, which one will be more? Plants or animals? Which is more? Plants or animals? What do you think? Is plants more or animals more? It's animals, okay? Just look at that. More than 70% of all species recorded are animals. Okay? And just 22% are plants. including algae, fungus, bryophytes, gymnosperms, angiosperms, that is just 22%. Okay, please underline those figures. 70% animals. Okay, 22% algae. Okay, fungus, gymnosperms. Okay, bryophytes, angiosperms. All these are just 22%. So just note this. And in animals, which one is more? In animals, which one is more? That opening paragraph, find out from the opening paragraph. In animals, which one is the most dominant? Insects. Okay, insects are very, very huge number. Okay, they have a huge number. Their diversity is very high. So insects, if there are 10 animals, 7 out of those 10 will be insects. Okay, so 70% will be insects in animal group. Got it? Now coming to that pie diagram, invertebrates. In invertebrates, you can see that insects are more, right? Just check that. Then comes other animal groups, then mollusca, and finally crustaceans. Got it? So we can learn it by some mnemonic. For example, IOMC. You can write India of my, what do you want with C? Put some word here. Okay, India of my C, coolness or something. Okay, just put something here, right? So what is this? Insects. Okay, other vertebrate and the invertebrate groups, mollusca and crustaceans. Okay, so the short form is IOMC. In the order, insects will come the most in invertebrate groups. Hope you understood this. Remember this mnemonic, you will be able to picturize this. This pie diagram, you're supposed to learn it by heart. Okay, they usually ask this, they'll give you the pie diagram. All those species will be missing. You're supposed to fill in the blanks. Got it, dear children? Hope you understood it. Yes? Shall we come to vertebrate groups? Shall we come to vertebrate groups? Make up a mnemonic. Then vertebrate groups. In vertebrate groups, you can use a mnemonic FB Ram. Okay, I think you will be able to remember that, right? FB Ram. Yes, what is F? Fish, right? Then comes birds, reptiles, amphibians, and mammals. Okay, FB Ram. This is fine. Very easy to remember the vertebrate groups. Check that pie diagram, everybody. Check the pie diagram. It's fine, right? Fish, birds, reptiles, amphibians and mammals, almost the same. Okay, this is okay. Amphibians and mammals, almost the same number you're going to find. Shall we proceed? Okay, the next one is plant groups. Okay, even fungus is coming under that category. Okay, they have put it under plants. Even fungus is grouped under that. You can put a mnemonic like this: AFAM. Okay, FL. That is, we'll just put a word like this. A for Arun. Okay, Arun is a fun guy. Okay, Arun is a fun guy. Okay, and Algin, another person. Okay, Algin makes fun of losers. Okay, Arun is a fun guy, but Algin makes fun of losers. Got it? So what is this? A is angiosperms. Okay, this is angiosperms. Fungi, fungi, right? Okay, algin. What is algin? Algin is obtained from algae, right? So that will be algae. Hope you understood it. Algae, right? Makes M for mosses. Okay, F for ferns, right? And these are lichens. Got it? Hope you understood it. Huh? Arun is a fun guy, but Algin makes fun of losers. You come up with some other mnemonic that you want. Huh? All these things are related with the plant itself, right? Hope you understood this. Check those three pie diagrams, everybody. Check the pie diagrams. Angiosperms and fungus almost same numbers you'll find. Okay, but if they ask you which is more, put angiosperms first because that's the most dominant vegetation that you're going to see now. Got it? Read that first paragraph and look at those figures. Everybody, do it right now. Do it right now. Write the mnemonic at the side so you remember it. And this is a 2-mark question. Okay, 2 marks. The pie diagram is a 2-mark question. Okay, is it done? Did you read that paragraph? Just one paragraph, okay? And look at those 3 diagrams. Check whether you have understood it. Shall we proceed? Shall we proceed, dear children? Okay, Devika, Surya. Iron, is it over? Ria, Felina. Okay, Aisha. So we are going to start the next one. See, if you look at eukaryotes and prokaryotes, all these what we are talking about are eukaryotes, right? It's very easy to understand or learn about the biodiversity in eukaryotes, but prokaryotes is a big problem. Okay, it's very difficult to find out the biodiversity in prokaryotic organisms. Main reason is they are very small. They cannot be easily cultured in the lab, right? So in order to learn about the biodiversity of prokaryotes is very, very difficult. Just check that last paragraph, right? Biologists are not sure about how many prokaryotic species there might be. Classification of these prokaryotic species is very tough because first of all, they are very small microscopic creatures. Second of all, most of them cannot be grown inside a lab condition. Okay, so all these are problems in classifying and finding out the biodiversity of prokaryotes. So that page is over. Page number 260 is over. Hope it is clear. Hope it's fine. Everybody. Okay. Next one. We are talking about India. Some numbers again are present there. Okay. See, India has, if you look at the world's total area, okay, world's total area, if you look at it, India has just 2.4% of the world's total area. Okay, but it the diversity, if you look at the species diversity, it is 8.1 percentage. So once again, India has just 2.4 percentage of the world's total area, but even though it has such a small land area, you have a huge diversity. The species diversity is nearly 8.1 percentage. Please learn these 2 numbers. Underline it right now. Hope you saw it. Page number 261, first line itself. Okay, and because of this, our country is among one of the 12 mega diversity countries in the world. Okay, so India is coming under the 12 mega diversity regions in the world. Hope you understood this. And if you are very interested to learn the numbers, there's a list of things that is found here. Can you see that? 1 lakh plant species, 3 lakh animal species, right? 45,000 species of plants, twice as number of animals has been recorded in India. Did you see those numbers? If you're very interested in learning numbers, you can give that a try. But they don't ask you that, okay? But just for the fun of it, if you like to by heart some numbers, there are a number of numbers there for you, okay? But remember this, these two numbers are very important. This is a question that is asked, okay? 1 mark comes for 1 mark. Got it? 2.4% of the world's total area. But if you look at the species, it's 8.1%. That's a very impressive number, right? So we are coming under the 12 mega diversity countries of the world. Okay, so that paragraph is over. Shall we come to the last paragraph for today? After that, we'll discuss the questions. So the next one, you can see patterns of biodiversity. Focus on the headings, okay? The questions will come, the headings are the questions. Got it? So what are the patterns of biodiversity? Okay, patterns of biodiversity. That could be a question. The first pattern of biodiversity, first one is latitudinal gradient. Okay, so the first one is latitudinal gradient. Okay, so in your geography classes, you might have learned the latitudes, longitudes, and all that, right? Hope you remember it. So this region, right? This region is a tropical region, 23.5 degree range, right? This is the equator. This is the tropical region. You have learned Tropic of Cancer, Tropic of Capricorn, right? Then comes the temperate regions, and then comes the polar regions. So if you look at the biodiversity, where will you find biodiversity highest? Will it be in the equator regions or will it be in the polar regions? What do you think? Where will you find biodiversity more? Is it at the equator or at the poles? Equator, right? Okay, you find biodiversity more in the equator. Got this? And in the tropical regions. These are the tropical regions. So this is the region where you find biodiversity the more. But if you travel further from the equator, as you move away from the equator, diversity is going to decrease. Is this clear? And there's an example given in your textbook. That example, please underline. See, Colombia is a country that is located very near the equator. And you are going to find 1,400 species of birds in Colombia. Do you see that? Okay, but New York is situated towards a temperate region. Okay, New York has how many species? Just 105 species of birds. And look at Greenland. Greenland is in the polar region, right? Just 56 species of birds are found. Hope you understood this example. So as we move from equator to the poles, diversity will decrease. Okay, the example once again, Colombia, which is located very near the equator, right? It's a South American country. It's very near to the equator. You have 1,400 species of birds. New York will come somewhere in the temperate regions, right? So you have 105 species of birds there. Greenland, which is coming in the polar regions, has just 56 species of birds. Hope you understood this. And India, they have given, India is also in the tropical region. We have 1,200 species of birds. Hope you understood this. Everybody has followed this. So when they ask you about latitudinal gradient, you can use this example: Colombia, New York, and Greenland. Okay, so just by heart those numbers. And if you want a bigger number, they have given Amazonian rainforests. Plenty of birds, right? You can see those 40,000 species of plants, 3,000 fishes, 1,300 birds, 427 mammals, 427 amphibians. I told you mammals and amphibians almost the same number. Okay. and 1,25,000 invertebrate groups. So such a huge number of species you're going to find in Amazonian rainforest. It's located in the tropics, right? Because of that, the diversity is very high. Hope you understood that. Hmm? And why do you think equator or tropical regions are having more species than compared to the poles? Is there a reason for it? Do you think, what do you think might be the reason? Equator is having, this region is having more species density when you compare it with the poles. What do you think might be the reason? Anyone can put a suggestion. What do you think?
Climate.
Somebody has unmuted themselves. Who's that?
Ma'am.
Felina. Felina, your mic is okay. Could be climate, right? BV. Climate is a big deciding factor. Anything else? Anything else? So shall we discuss the reasons? There are three reasons. Okay. So the question is why tropics have high species density compared to the poles. Okay, so why do tropics have high species density when you compare it with the poles? That is our question. The first reason, see, when there was when the earth was evolving, right, there were periods of glaciations. You might have heard about ice ages. If you have seen that movie, now we need more exposure to sun. Yes, we're going to discuss it. If you have seen the movie Ice Age, you know about glaciations. Have you seen that movie? Watch that movie, Ice Age? Yes, Surya? There's 1, 2, and 3, right? The first one is the best episode. Hmm? Yes or no? You might be familiar with glaciations, ice ages. So if you compare the polar regions, there were more glaciations than in the tropical regions. Okay, so that was the first reason. The polar regions have faced more glaciation. So what will happen? The entire species present at that time will be wiped out. Okay, new species have to be formed. But temperate regions were more or less stable. Got it? That is the first reason. And the second reason could be, see, if you take a tropical regions, our seasons are almost the same, right? We have one rainy season and one summer season. That's it. But if you look at a temperate regions, you find a wide range of seasons. Hope you understood this. There's winter, there's summer, spring, autumn, all those seasons are present. So it is much better for species can adapt much better in a tropical region than a temperate region because there's a seasonal fluctuation that is happening. Tropics, they have a much stable seasons. Okay, most of the time it's a hot climate, right? There's no much of a fluctuation that is happening, but in tropics you find seasonal fluctuations, so it's very difficult for a species to adapt. Got it? That's the second point. And the third point is in tropics you have maximum solar energy, right? The solar input is maximum in the equator regions. So what happens if solar energy is maximum? The productivity, right? The plants can use that energy and produce food maximum in this region. So animals can flourish in that region more. The diversity will be more there. Hope you understood this. 3 points. Yes, this is a question that is constantly repeated. Okay, it's again a 2-mark question. This is again a 2-mark question. Got it? So just read that and we'll wind up for today the chapter. Okay, then after that we'll discuss the questions. So just go through it. Take a latitudinal gradient, read it word by word. Underline these things. First point, glaciations. In the poles, glaciations are more. Okay. And in temperate regions, glaciations were less. Glaciation means ice age. Yes, Felina, got it? Felina, did you understand that first point? Speciations are generally a function of time. Unlike temperate regions, subjected to frequent glaciations in the past, tropical latitudes have remained relatively undisturbed for millions of years and had a long evolutionary time for species diversification. Okay, and ask me a doubt after that. After you read it, ask me a doubt. There's one word you should be asking me a doubt. Let me see who finds that word. Okay, did you find some doubt, dear children? Did you find something to ask me? There's one word that you should have a doubt. Nobody has got it? Ah, BV, good, good. Okay, niche specialization. Did all of you see it? Niche specialization. Okay, this is a word that is given in your textbook. Huh? See, in the second point, they have written like this. Tropical environments, unlike temperate ones, are less seasonal, relatively more constant and predictable. Such constant environments promote niche specialization and leads to greater species diversity. Okay, so because there's a constant environment, what will happen? Those species will be able to adapt themselves better to the biotic and abiotic factors. Okay, so this adaptation of a particular organism to its biotic and abiotic factors is called niche specialization. They are able to live in a specialized environment. Okay, is this fine? Everybody, they are able to adapt themselves to the specific biotic and abiotic factors found in a specific region. It's called niche specialization. Is this fine? Everybody has got it? Just note it down, okay? But if the temperature and climatic factors are changing, it's very difficult for the organism to adapt itself, right? Is this okay? So what all did we learn today? We'll just wind it up. We have learned biodiversity. We have learned a few examples for biodiversity. How many ant species, how many orchids, right? How many beetles. Those numbers are very important. We have talked about Edward Wilson. According to him, Edward Wilson was the person who popularized that term biodiversity. Right? According to him, there are different levels for biodiversity. One is a genetic level, the second is a species level, and the third is the ecosystem level. Right? And the examples of genetic level, species level, and ecosystem level are very, very important. You have to learn those examples. Now coming to how many species are present, according to IUCN we have seen a number, according to Robert May we have seen another number, right? If you turn the page, you'll find 3 pie diagrams. Those 3 pie diagrams you're supposed to by heart it just like that. You'll sit and this will be your homework, okay, for today. Draw those pie diagrams and label it. If you look at plants and animals, animals are more, plants are less, okay. 70% and 22% is the number. If you look at animals, insect numbers are more, okay. And they've given 3 pie diagrams, one for invertebrates, one for vertebrates, and the other for plants. We have used a mnemonic. If you have a better mnemonic, you can make that and learn it. This is fine. Next one, we have seen that prokaryotic species is very difficult to identify. Its diversity is very difficult to note, okay. And we have talked about India. It has just 2.4% of the world's total area, but 8.1% of species diversity, you find it here. So we are coming under the 12 mega diversity rich regions in the world. Okay, then we have talked about patterns of diversity. The first pattern of diversity we talked about was latitudinal gradient, right? If you go from equator to the poles, what will happen? Diversity is going to decrease. And that example of Colombia, New York, and Greenland, you have to remember. Okay, and Amazonian rainforest, if you can by heart those numbers, it's okay. Once they had asked this for entrance, those numbers were asked for entrance. Okay, that's very rare, but still, if you can remember it, well and good. And what is the reason for this? Why are tropics having species density, more species density compared to the poles? This will be your second homework for today. Okay, this will be your second homework. Why are tropics having very high species density compared to the poles? You are supposed to write 3 reasons. Hope this is clear, dear children. And Solomon, did you understand that absence of fingerprint? Which fingerprint were you talking about? Did you understand that? We are not talking about of thumb impression, okay? We are talking about DNA. Solomon, are you there? Hope all of you understood it. I had put it in the group just after the class. Did you read it? You were talking about the thumb, I was talking about the DNA, right? We were talking about 2 different things. Okay, hope you understood it. Hope this is fine. So shall we do the questions? Shall we do the questions? Take question number 31. Question number, biotechnology applications. We have discussed up till question number 30. We need to discuss question number 31. So take question number 31. Okay, which one of the following pairs or terms means one and the same thing? Okay, which is your answer? Put your answer key right now, everybody. Okay, D, right? D is DNA fingerprinting, or it is also called DNA profiling. And cybrid is cytoplasmic hybrid. Okay, so just note down this word, cybrid. Cytoplasmic hybrid. A hybrid that is formed by the fusion of cytoplasm but not the nucleus. Okay, so just note down this word, cybrid. Okay, what is a cybrid? Cytoplasmic hybrid. A hybrid that is formed by the fusion of cytoplasm. Okay, and not the nucleus. Okay, but that is not a clone, right? Then next one, transgenic organism is called. What do you call a transgenic organism? Put the options. C, right? GMO, genetically modified organisms. That is a common name that you can use. GMO, genetically modified organism. Okay. Then in transgenics, the expression of trans gene in a target tissue is determined by its promoter. Okay, the promoter regions. Promoter is from where the transcription is going to start. Got it? Okay, next one. genetically modified brinjal in India has been developed for? GM brinjal is for? We have already talked about BT cotton, just like that, BT brinjal. Okay, insect resistance. We have seen lepidopterans, dipterans, and coleopterans. Larvae can be killed by BT. Right? So option A. Next one. Bacillus thuringiensis stains have been used for designing novel D, right? Bio insecticidal plants. Iron, you need to participate in the discussion. Everybody will put your answers there, okay? Read the question, open your textbook, keep it with you. Iron, please participate in the discussion. Everybody. Jagannath. Where's Jagannath? Jagannath, you're there, right? Put your answers. Let it be wrong. Nobody is going to judge you here, right? Just put your answers. Whatever you feel, just put the answers, right? We'll discuss the right one. So 35 is D, right? BT we have seen, it's for insecticidal properties. Okay, BT toxin kills the larvae of certain insects by, we've already seen it. Felina, Surya, good. Let's see who will answer first, right? Okay, by binding of activated toxin on the midgut of epithelial cells, creating pores leading to swelling and lysis. Okay, a very evil way to kill an insect. Okay, then transgenic plant, flavor saver tomato, carries an artificial gene for, we have already talked about it. It could be D, right? Okay, longer shelf life. Carries an artificial gene for, okay, all of these. It could be all of this, D. Okay, then Dolly sheep was genetically similar to. Okay, Dolly, that was the first clone, right? Ian Wilmot, you might have learned about him, Ian Wilmot. So what they did was, they took a cell and this was enucleated. This was the enucleated udder cells. From the udder of the sheep, they took a cell and this was made enucleated. The nucleus is removed. And then from a donor sheep, the nucleus was implanted. So this nucleus is from the donor sheep. Got it? So see, they are combining two things together, right? The cell is from one, the first sheep. Okay, they have taken a nucleus from a donor sheep and they have implanted it inside, and then they have fused it. So whose character will this dolly have? Whose character will the dolly have? Will it be of this, the one with the enucleated cell, or the donor? Right, the mother from which the nucleated udder cell was taken, the nucleus was taken, right? B. Is this fine? The nucleus contains the chromosomes. So whichever cell has a nucleus, that character will be shown by the offspring. So B is the answer. Okay, next one. A technique of gene transfer using very small metal particles coated with desired gene is referred to as. Surya, good. Yes, Felina, Devika. The girls are winning. Okay, none of the boys are coming first. Solomon, Jagannath, Navneet. Pick up pace. Okay, see, right, biolistics or gene gun method. I have already sent you that video. Hope you have observed it. Okay, 40. Blindness can be prevented by the use of which crop in poor countries? Blindness can be cured by the use of which crop? See, wherever there's vitamin A is very good for your eyes, right? So a crop that is rich in vitamin A, we have already talked about golden rice, beta carotene content, vitamin A content is very high. See, blindness because of lack of vitamin A, xerophthalmia. I think you're familiar with it, right? Golden rice is the answer. Xerophthalmia is the disease. Okay, so B. 41. Transgenic pigs are usually applied in the field of. Where will you find transgenic pigs? See, it's usually in organ transplantation. Human and pigs, okay, the organs are very similar. See, the valves, heart valve, all those are taken from pigs. Okay, they have very similar organ composition. Got it? So they are used in organ transplantation. Okay, 42. Select a statement which is not related to transgenic tobacco. Select a statement that is not related to transgenic tobacco. I think all of them are related, right? All are related. Yes, I think I have written that answer there. Okay. Then 43. Maximum number of existing transgenic animals is of. You know that, right? Mice. Maximum number is that of mice. Now, GEAC. What is GEAC? C, right? C. Genetic Engineering Approval Committee. They look at two things. Just put the two things that GEAC look into. Let me see who answers first. Who will answer first? Validity and BV good. Validity and one more word. Safety. Good BV. Validity, productivity. Surya, it's not productivity. Validity and safety of genetic research. Right? We are not talking about inbreeding depression. Okay, Surya? Next one. How many varieties of rice have been estimated to be present in India? See, I think in two different places there are two different numbers, right? According to this chapter, it is which option? C, right? C or B? C, right? Okay. Then 26. Gene therapy can be referred to as. What is gene therapy? That's B, right? Treatment of a disease caused by a genetic defect. You are putting in a gene, a functional gene into the body, right? The first disease to be cured, ADA, adenosine deaminase deficiency, right? Name of the girl, do you remember? What was the name of that girl? The 4-year-old child. Do you remember her name? Nobody remembers the name. It was a very weird name, right? Nobody remembers the name? Huh? Name of that 4-year-old child. I think I mentioned Ashanti, right? Ashanti De Silva. That was the name of that child. Okay. Then gene therapy can be referred. Sorry, Hirudin. Hirudin is. See, Hirudin is an anticoagulant that leeches will produce normally. Okay, Hirudin. Because leech is called Hirudinaria. I think we have learned it last year. It produces an anticoagulant when the leech, right? In Malayalam, you call it atta, right? When it is drinking your blood, the blood will not clot. It injects a anticoagulant into your skin, so your blood will not clot until it has finished drinking, right? And that creature will be very small in the beginning. After drinking blood, it will start swelling up. You're familiar with leech, okay? And it produces the chemical hirudin. So we are artificially producing that. We have put an artificial hirudin gene in Brassica napus. Okay? Brassica napus, it's oil seed. And then we are producing that chemical hirudin from Brassica napus. So your answer is C. A protein produced from transgenic Brassica napus, which prevents blood clotting. So you're making hirudin protein in a plant. Got it? So it's a transgenic plant. C is your option. 48. Main objective of production of bioherbicide resistant GM crop is to. What do you think is the main function? Don't you think D will be a better option? Navani, D is a better option, right? To eliminate weeds from the fields without the use of herbicides, because a plant without the use of herbicides. right? I think D would be a better option. Huh? Let me check that once again. Bioherbicide resistant GM crops. Okay, we'll come back to that question once again. Okay, let me just check once again. 49. In tomato, the softening of middle lamella is caused by the enzyme. Which is that enzyme involved? I think in the first question we have already discussed that enzyme. D, right? Polygalactouronase. Felina, yes. Navneet, okay. D, polygalactouronase. It will weaken that cell wall. Pectinase degrading enzyme. Okay. Okay, 50. Adenosine deaminase deficiency results in which disease? Adenosine deaminase deficiency results in which disease? Which disease? You can put it as SID, okay? Severe combined immunodeficiency. SID. Okay, just write the full form. Severe combined immunodeficiency. If you have seen, there's a movie based upon this. He's called The Bubble Boy. There was a boy who was he was born with this disease. That means his immune system is not at all functioning. Okay, he cannot come in contact with other people. He used to live in an artificial bubble. Have you seen that movie? It's called The Bubble Boy or something. It's famously called The Bubble Boy. He used to live inside a sterilized bubble. Huh? Okay, that's SID, severe combined immunodeficiency. Even this one is affecting your lymphocytes. So your deficiency will be affected, sorry, your immunity will be affected, right? So put it as C. 51. Which of the following genes were introduced into cotton to protect it from cotton bollworms? It's there from in your textbook, right? C, option C. CRY1AC and CRY2AB. Yes. 52. Which of the following statements are correct? Which of the following statements are correct? Read all the statements. It's kind of a revision for you too. Which statement? BV, Aisha, D, right? Iron, good. It's D. All statements are true. Okay, read 53. Which of the following statements are correct? Read it thoroughly, okay? Maybe one word will be wrong. D, right? All the above are true. Okay. 54. Select the correct statement. Select the correct statement. C. First step in PCR is to heat, which is used to separate. So what is that step called? Everybody name the step. The first step of PCR. Put the first step. Everybody, you're heating it 94 to 96 degrees Celsius for a few seconds. What do you call that step? What do you call the first step of PCR? Where did everybody go? Riya, Aisha, Devika, Surya, Navaneed. Denaturation, right? It's called denaturation. Second step is called? Second step, when you're adding the primer, you call it? Annealing, right? And then you are adding DNA polymerase, stack polymerase. That step will be called extension. So there are 3 steps, and temperature, hope you are familiar with it. For extension, what is the temperature? It's very important. For extension, what is the temperature that is required? Let me see who answers first. You're turning the pages, I believe. What is the temperature required? Everybody is busy turning the page. Nobody knows the answer. Let me see who finds it out first then. What is the temperature for extension of DNA? I've given the temperature. It's not there in the textbook. BV 72. Right? 72 degrees Celsius you have to use. Okay. Then next one, 55. Human insulin is being commercially produced from a transgenic species of. We know that. We know that answer. Human insulin, E. coli, right? The first company was Eli Lilly. The year was 1983. Okay, next one. The linking of antibiotic resistance gene with a plasmid vector becomes possible with the help of linking. D, right? DNA ligase. You can link it using DNA ligase. It will seal the gaps. Okay. XIIE2AB and XIIE1AB produce toxins that control. Yes, Felina, good. Option A, cotton bollworms and corn borer. There should be a competition. Amulia, good. Priya, good. Okay. Let's see who will answer first. Almost 95% of all existing transgene animals are E, right? Mice. Okay, good. Who is the first one? Surya, good. Transgenic models exist for which of the following human diseases nowadays? E, right? Okay, Surya again. All diseases, cancer, I told you oncomice, cystic fibrosis, rheumatoid arthritis, Alzheimer's disease, all these models are present. So E. 60. Which of the following is a wrong statement? Which of the following is a wrong statement? Devika. Is it Devika who answered first? E, right? I think the year is wrong. Which is the year? In which year? 1997, right? They have written 1990. Okay. Now 61. I think 61 I've sent you yesterday from 61. Which of the following BT crops has been grown in India by farmers? Which was a more successful BT crop? A, right? BT cotton. Yes. Genetic engineering is possible because. Why is genetic engineering possible? We have seen the first tool of genetic engineering, right? Restriction endonucleases. Because of restriction enzymes, we are able to cut it. Solomon, Jagannath, are you there? You're not putting the answers. I cannot see it. So A, which of these is used as a vector in gene therapy for SID? That is for ADA deficiency. We are using some, which one? Retrovirus. It's there, right? We have learned lymphocytes were extracted from her body into the lymphocytes. Functional ADA gene was was introduced using a vector. That is a retrovirus. Okay, so C. 64. During the process of pro-insulin into mature insulin, processing of pro-insulin into mature insulin, what should happen? Surya, good. Surya is the first one to answer. Okay. C peptide is removed. Hope you remember the structure. There was A peptide, A and B peptides joined together by a C peptide. If you remove the C peptide, it becomes mature insulin. There are disulfide bonds between. Okay, then this is the next one, 65. BT toxin is not toxic to humans because. BT toxin is not toxic to humans because. What will be the answer? C, right? C is your answer because in order to make that toxin active, you need alkaline pH. In human stomach, it is acidic pH. So C. 66. Find the answer. Arrange the steps in the correct sequence and select the correct answer. 66. What is your answer? A, right? Option A. It's option A. Okay, 66, put it as option A. Felina, you're correct. Next one, 67. Interferons are useful in. When will you use interferons? Interferons, we have seen it as certain special proteins that are produced by cells infected with virus and they are messaging other cells, right? We have talked about cytokine barrier of immunity. So C is the answer. Next one. First transgenic plants was produced in tobacco. Okay, now 69. The main objective of production use of herbicide resistant GM crop is to. Same question. Okay, we'll just come back to that question again. 70. What is the disadvantage of using porcine insulin in diabetic patients? Porcine insulin means from pig. If you're taking the insulin of the pig and injecting it into human, what will be the problem? It may cause allergy, right? Because it's a foreign product, it might cause an exaggerated immune response. That's an allergy. Okay, 71. Which technique would be completely curative in SID? Which technique would be completely curative in SID? B, right, Navneet? It's B. Gene therapy in embryonic stage. If you take a mature cell, what will happen after a few days? 3 to 4 days, those cells will die off. So you need to do that treatment again. But if you do it in the stem cells in the embryonic stage, it will last you a lifetime. Got it? So B is your answer. 72. Through Green Revolution has been a resounding success in terms of agricultural production, yet it has failed in its overall social objective because D, okay, option D, all of these. 73. Which of the following statements regarding gene therapy is correct? Read the statements and find out which of the following statements are true. It's D, right? Okay, all statements are true. Okay, 74. Which of the following statement is correct with regard to disadvantages of GM crops? Disadvantages of GM crops. 74. Put the answer. Read it, okay? If you have not done it, just read. Read and tell me. Yes, what is the option? 74. Surya, D, right? Option D, all of these. 75. Genetically engineered bacteria have been successfully used in commercial production of human insulin. You call it humulin, right? Hope you remember the steps one by one. Then 76. Bt toxin shows specificity to. Bt toxin is highly specific to. Is it fish, insect, mammals, birds, protozoans? B, right, insects. Which all types of insects? Coleopterans, dipterans, and hemipterans, right? Yes or no? So 3 types of species. And you are supposed to learn the examples of that also. Which are the dipterans, which are the coleopterans, and all those things. Then 77. Which of the following gene is responsible for production of Bt toxin for the control of cotton bollworms? Which one? 78? Sorry, 77, right? 77. E, right? Both B and C. B is CRY1AC and CRY2AB. By heart it. Then 78. Cry genes have been introduced in. 78. It can be D, okay? In all of them, it has been introduced. Got it? D. 79. Bt toxin is harmful to insects like? It's harmful to insects like? 79. D, right? Option D. Okay. See, did you learn those examples? Lepidopterans, tobacco, bollworms, right? Army worms, coleopterans, beetles, dipterans, flies, and mosquitoes. Learn the examples of all these things. Okay, last question. Golden rice developed through transgene approach is enriched with D, right? Vitamin A content. Okay, so we'll do one thing. See, I'll send the rest of the questions from this chapter. There are 105 questions. Okay, there are 105 questions. So we'll do it in the next class too. So what will you do today? There are 2 homeworks for you. Read up till that properly and prepare for the examination. Okay, that whole unit 8, biology and human welfare will be your unit for the examination. Got it? So just go through it, see if it's fine. Any doubts from today's session? Any doubt? If so, we can wind up. Any doubts, dear children? Is it fine? So shall we wind up today's class? Okay. So thank you. Thank you, everyone.